AppSec's online support program, Class 8, Week 14, Subject Biology, prepared by Shaista Zeeb. Textbook Biology 9, Lahore Publishers. Pages number are 87 up to 88. Chapter, Unit, Cell Cycle, Unit 5. Lesson topic, Cell Cycle, Interface, Resources required, Textbook, Internet connection, Notebooks. Introduction summary of the lesson. This lesson is about the cell cycle and its phases, interface and its further phase. At the end of this lesson, you will be able to know about cell cycle, two phases of cell cycle, that is mitotic phase and interphase. Interphase subdivision, G1 phase, S phase, G2 phase, G0 phase. Recap. In the previous chapter, we studied about the discovery of cell structure and function, cell organelles and its function and composition. Dear students, today our topic, that is cell cycle. What is cell cycle? The cell cycle or cell division cycle is the series of events that take place in the cell that cause it to divide into two daughter cells. This event includes the duplication of its DNA and some of its organelle and subsequently the partitioning of its cytoplasm and other component into two daughter cells is called the cell division. Cell division is the series of events from the time cell divides produced until it completes mitosis and produces new cells. Cell cycle occurs from one cell division to another cell division. All cells are derived from pre-existing cells. New cells are produced for the growth and to replace damaged or old cells. Cell cycle consists of two major phases, that is interphase and mitotic phase. Let's see in the flow chart, they are subdivided into interphase and mitosis phase. Mitosis phase are further divided into karyokinesis and cytokinesis. Karyokinesis is further divided into prophase, metaphase, anaphase, telophase. Dear students, this is the schematic of the cell cycle. The eukaryotic cell cycle consists of four distinct phases: G1 phase, S phase, G2 phase, collectively known as interphase, and M phase, mitosis and cytokinesis. M phase is itself composed of two tightly coupled processes: mitosis, in which the cell nucleus divides, and cytokinesis, in which the cell cytoplasm divides, forming two daughter cells. Activation of each phase is depend on the proper progression and completion of the previous one. Cells that have temporarily or reversibly stopped dividing are said to have entered a state of consequences called G0 phase. Cell cycle. All eukaryotic cells store genetic information in chromosomes. Most eukaryotic have between 10 to 50 chromosomes in their body cells. Human body cells have 46 chromosomes or 23 identical pairs of chromosomes, and we discussed about the chromosomes in our previous chapter, that is cells and tissue. This is the diagram of the chromosomes. This is the microscopic picture of the chromosome. Interphase. Interphase is a series of changes take place in a newly formed cell and its nucleus before it becomes capable of division again. It is also called preparatory phase or intermitosis. Typically, interphase lasts for at least 91% of total time of cell required. Interphase proceed in three stages, that is G1, primary growth phase, S, synthesis phase, DNA is replicated, G2, secondary growth phase. Collectively, these three are called interphase. Interphase G1 phase. Its first growth stage after cell division. The first phase within interphase from the end of the previous M phase until the beginning of DNA synthesis is called G1. G indicate gap. It is also called the growth phase. During this phase, the biosynthetic activities of cell, which are considerably slowed down during M phase, resume at a high rate. The duration of G1 is highly variable even among different cells of the same species. Cell mature by making more cytoplasm and organelles. Cell carries on its normal metabolic activity. Interphase S phase. S means synthesis stage. In this phase, cell duplicates its chromosome. As a result, each chromosome consists of two sister chromatids. DNA is copied or replicated. This is the original DNA. Two identical copies of DNA will form. And that in this stage, in the interphase, it will divide into two identical copies of the DNA. Interphase G2 phase, second growth stage, occurs after DNA has been copied. G2 phase occurs after DNA replication and is a period of protein synthesis and rapid cell growth to prepare the cell for mitosis. During this phase, microtubules begin to recognize to form as spindles. All cell structure needed for cell division are made. For example, centrioles. Both organelles and proteins are. Dear students, let's see what's happening in the interphase G2 phase. What the cells look like. Animal cells. Look at the animal cell with a full label diagram. What occurring? DNA replicates, centrioles are present replicate, cell prepare for cell division. Interphase G0 phase. G0 phase is resting phase where the cell has left the cycle and has stopped dividing. The cell cycle starts with this phase. Cells that have temporarily or permanently stopped dividing are said to have entered a state of G0 phase. Eukaryotic cells enter G0 phase from G1 and stop dividing. Some cells enter G0 phase for indefinite period. For example, neurons. Some cells enter G0 phase semi-permanently. For example, some cells of liver and kidney. Some cells never enter G0 phase like epithelial cells. Written work. What is the cell and what are its main phases? Question number 2. The S phase of interphase is important and a cell can never divide without it. Justify. Thank you, dear students. Have a good day.